Okay, so hi guys. Assalamualaikum warahmatullahi ta'ala wabarakatuh. Greetings of peace. I am Muhammad Nasif Batugan, ating a first year Bachelor in Arts Psychology major student, and I am here for I was tasked to discuss and report to you the chapter 12 of our topic, which is all about the ribonucleic acids in the brain. So let's start decoding it first. What is nucleic acid? Since RNA or the ribonucleic acid is a nucleic acid. So, nucleic acids are naturally occurring chemical compounds that serves as the primary information carrying molecules in the cell. So, they play an especially important role in directing protein synthesis. So, its role is it serves as information carrying molecules in the cell. So, they play an important role, for they are the one that makes protein synthesis, or that creates protein. So there are two main classes of nucleic acids: the deoxyribose nucleic acid and the ribonucleic acid. So the deoxyribonucleic acid is the one that we call DNA, and the ribonucleic acids are the one that we know, the RNA. So let's start with what is RNA. So RNA is ribonucleic acid, so it is a long single strand of chain cell that processes protein. So unlike DNA, which is a double stranded, RNA is a single stranded chain of cells. So it is a polymeric molecules essential in various biological roles in coding, decoding, regulation, and expressions of genes. So aside from its main role, which is to process protein, it also serves as it also has biological roles, for it codes, decodes, regulates, and expresses the genes. So RNA is present in all living things, not just in human cell. So its principal role is to act as the messenger carrying instructions from DNA for controlling the synthesis of the protein. So later on we'll discuss on that one. So here I have the differences between the RNA and the DNA for us to be able to fully understand what's RNA. So first difference is that, as I said kanina, RNA is a single strand while DNA is a double stranded. So for its sugar, RNA's main sugar is the ribose, while for the DNA it is the deoxyribose. So for its nitrogenous bases and pairings, so pyrimidine components differs. So for the DNA, thiamine is never found, while for the DNA, thiamine is always there, but uracil is never found. So for the base pairing of the RNA, we have the guanine is paired with cytosine, and adenine is paired with uracil, since thiamine cannot be found and never found in the RNA. While for the DNA, we have guanine is paired with cytosine, and adenine is paired with thiamine, since uracil cannot be found in the DNA. So RNA does not follow Chargaff's rule, and for the DNA it follows Chargaff's rule. So number 5, we have here RNA can easily be destroyed by the alkalis to cyclic diester of mononucleotides, while the DNA can resist to alkali action due to the absence of the OH group at 2 positions. So RNA can be destroyed by the alkali, while the DNA can resist to alkali. So for the number 6, we have here RNA are relatively liable molecules, which means that they are always on move, while the DNA is a stable molecules, for it can only be found inside the nucleus. So RNA is mainly cytoplasmic, for it can be found outside the molecule, and it is always on move molecule, while the DNA, for it was a stable molecule, again it can only be found in the nucleus. So we have here RNA content varies. The base content of the RNA varies from 100 to 5,000, while the base content of the DNA are million base pairs. So for the RNA, it has various types. It has the mRNA or the messenger RNA, the ribose RNA, which is the rRNA, and the tRNA, which is the transfer RNA, and many more. So while for the DNA, it only have one type. DNA is always DNA, unlike RNA that it is subdivided into different subcategories. So for the DNA is DNA. So it only has one type. So for the number 10, we have here RNA has no variable physiological forms that can be found, while for the DNA, it has variable psychological forms from A to E and the Z. So for the number 11, RNA is synthesized from DNA and it cannot form DNA. So RNA is created by the DNA. So for the DNA, it can form DNA. DNA can create DNA by means of replication, and DNA can also create RNA by means of transcription. So for the last differences we have here RNA. Many copies of RNA are present in the cell, while single copy of DNA is present per cell, which can only be found inside the nucleus. Okay, so now let's start decoding how the DNA and the RNA works together. Since both of these nucleic acids plays vital role in creating protein or in synthesizing protein, they need to work together. So the DNA is the one that has the information, or that creates the information how to synthesize protein, or how to create protein. So the information is all in the DNA. So for it to go, or for the information to go to the ribosome in the cytosol, it needs to make RNA. So since the DNA plays vital role, it cannot go outside the nucleus. For inside the nucleus, it is protected. So it needs to stay inside the nucleus. And for the information to go outside the nucleus, the DNA creates RNA by means of transcription. So it creates DNA creates RNA. For the RNA is the one that will go to the ribosomes in the cytosol. So now we have here the types of the RNA. So in all prokaryotic and eukaryotic organisms, three main classes of RNA molecules exist. So these are the three main classes: the messenger RNA, which is also known the mRNA, the transfer RNA or the tRNA, and the ribosomal RNA, which is the rRNA. So aside from these three, we also have other types. We have the small nuclear RNA, microRNA, the small interfering RNA, and the heterogeneous nuclear RNA. So we'll start decoding what does each of these type of RNA makes. So let's start with the messenger RNA. So messenger RNA is a single strand that carries messages from the DNA in the nucleus to the ribosomes in the cytosol, so that it can be used to make protein. So ayan, the DNA is the one that has the message. So the messenger RNA is a single strand that carries this message from the DNA or inside the nucleus going to the ribosome. So the ribosome is inside the cytosol. So this information or this message are that the DNA gives to the RNA is the information on making protein. So it comprises only 5% of the RNA cell, and messenger RNA is mostly heterogeneous in size and base sequence, and all members of the class functions as messengers carrying the information in gene to protein synthesizing machinery. So next one we have here the transfer RNA, which or the tRNA. So it is a clover leaf shaped single strand that carries amino acids to the ribosomes and helps to assemble them in correct order. So after the messenger RNA sends the message to the ribosome, transfer RNA gets to work. So what does it do? So it carries amino acids. So these amino acids are being transferred to the ribosomes from the nucleus, and these amino acids are the one that will be or are the one that is going to be protein. So aside from transferring amino acid, it also transfer RNA also helps to assemble these amino acids in correct order for it to be able to make correct protein. So transfer RNA are the smallest of three major species of RNA molecules. So they have 74 to 95 nucleotide residue. They are synthesized by the nuclear processing of a precursor molecule. They transfer the amino acids from cytoplasm to the protein synthesizing machinery, hence the name tRNA. They are easily soluble, hence they are called soluble RNA or the sRNA. So lastly, they are also called adapters molecules since they act to adapt for the translation of sequence in nucleotides for the mRNA into specific amino acids. So for the last type or the last main type of the RNA, we have here the ribosomal RNA. So it is a single strand in globular form. So our RNA binds with protein to make up ribosome, which are then used to make protein. So the messenger RNA gives the information to the ribosome, then the transfer RNA gives the amino acid to the ribosome, and the rRNA binds with protein, and then it makes up ribosomes which are then which then becomes protein. So that's how protein works, or that's how the DNA and the RNA works together. So another additional information about the ribosomal RNA, the mammalian ribosomes contains two major nucleoprotein subunits, a larger one with molecule weight of 2.8 times 106/60s, and a smaller subunit with a molecular weight of 1.4 times 106/40s. So the 60s subunit, or the larger one, contains a 5 ribosomal RNA, a 5.8s RNA, and a 28s rRNA. They are also probably more than 50 specific polypeptides. So the 41, or the smaller subunit, contains a single 18s rRNA and approximately 30 distinct polypeptide chains. So the bigger one has more than 50 specific polypeptides, while the smaller one has approximately 30 distinct polypeptide chains. So all of these ribosomal RNA molecules except the 5s RNA are processed from the single 45s precursor RNA molecules in the nucleus. So the 5s RNA is independently transcribed. So let's move on now to the RNA synthesis, or the RNA transcription, wherein how RNA is being made from the DNA. So RNA synthesis or transcription is the transfer of information from the DNA, where it is stored into an RNA which can be transported and interpreted. So again, three major classes of RNA synthesized by transcription of appropriate genes and involved in protein synthesis. We have messenger RNA, ribosomal RNA, and the transfer RNA. So messenger RNA carries the message from the DNA to the ribosome. Ribosomal RNA, major structural component of protein synthesizing ribosome, and last one, the transfer RNA, act as adapter molecules in aligning amino acid to according to the sequence present in the messenger RNA. So DNA dependently RNA polymerase, which is the nucleotide triphosphate, are responsible for synthesizing of cellular RNA. So they bind to DNA and polymerize ribonucleotides into RNA molecules. So the primary products of RNA polymerase activity are a very large polymers, so much larger than average messenger RNA molecules, and they are present in the cell nucleus and are termed heterogeneous nuclear RNA. So that's the product of the RNA polymerase activities. It creates a large polymers. So now let's go to the gene expression in neurons. So in terms of diversity, brain messenger RNA is about 4 times as complex as the RNA in the liver. About 16 to 20 of unique DNA sequence is transcribed in the adult rodent brain. Estimated this value at 31% as compared to the 10% of the kidney. So studies of gene expression in neurons are still hampered by gaps in our general understanding for the gene control in eukaryotic cell. In this cell, the phenomena of gene expression includes the loss amplification and rearrangement of gene materials, changes in nucleosome structure, and DNA methylation. So there is an involvement of various nuclear proteins, both histone and non-histone, some of which can serve repressors or activator capacity. So now let's go on to the brain RNA. So the synthesis and metabolism of RNA in adult nerve is known to be responsive to neuronal functioning. Thus, the stimulation of peripheral receptors and consequent sensory activities increases the synthesis of ribonucleic acids throughout the entire sensory pathway. So RNA data are often difficult to interpret, since one must take down into consideration a number of influential variables, including the rate of RNA degradation, the accessibility of precursor, and availability of the energy supply. So brain RNA, there is presently no evidence showing that increase in RNA synthesis in neuron is a specific response to the learning processes. Experiments with simple system or with sensory stimulation indicates that sufficiently intensive postsynaptic excitation can itself elevate RNA synthesis. These effects involves the various forms of RNA, which is the nuclear and cytoplasmic, ribosomal RNA, the messenger RNA, and some transfer RNAs. So the RNA polymerase activity. And lastly, the role of ribonucleic acids in learning and retention has also been studied by the administration of drug which inhibits or stimulates RNA synthesis. Some of the stimulating substances formerly hailed as memory improving drugs whose effect were attributed to raise in RNA protein synthesis did not warrant an enthusiasm which they had been welcome. So that's it. Assalamualaikum warahmatullahi ta'ala wabarakatuh. Thank you everyone.